So this will be the second lecture about more details about the place cells, and then also more details about how they actually, what are some models about how they actually might work, because as somebody rightfully asked last time, it just doesn't, how could this possibly work? So we'll start off just with that first, and so what's happened has just been an absolute explosion of experiments on place cells and head direction cells and grid cells. And there's a Nobel Prize, a couple of Nobel Prizes, awarded for this. But that first experiment I was talking about was you have this arena, and it's got some kind of cue card in it, like this, just to sort of indicate to the animal where it is, in the arena. And then you got the animal just running around. So there's a little rat, and it's just like it's just sort of running all around in there. And then you're recording. You got the camera, and you're just recording where the rat is. And what is the basic, very most basic observation, just to review? And the most basic observation is that we've got, if we look at it from the top, so this is top view. So we look at it from the top and just record where the spikes occur in XY. So what are we looking at here? We're looking at, spike rate as function of position. So basically, when a spike occurs, we just paste it down in our diagram where it occurred. And if you do that, what you see is, say the cue card is sort of sitting here, like that, in that circular arena. What you'll see is, the spikes will occur like in one place in the arena. There'll be a few spikes here and there, but basically 97% of the spikes will occur just in that one place. And so that's the place field. So this is our place field. And like when this was first observed, a lot of people, like me, just said, "Nah, the brain doesn't work that way. That can't be true." But it is true. And so what is the basic observation that shows that place is related to these, what we'll call distal cues? And that observation is, you do this, takes about 10 minutes to do this, 5 or 10 minutes, record those spikes. Obviously, you have to somehow make the animal go to all different places in the environment, like I said, by checking in some rat chow so that they're sort of exploring the environment. But then you take them out. So now we, take them out, and then we confuse a rat. So we take the rat out of the container, and we sort of move the container around a little bit, maybe go in another room, keep the container closed so they can't see, where they've gone. I always say it's like rat rendition. You grab a rat and turn them up, and they no longer know, exactly where they are oriented relative to the environment, and then put them back into the arena. And usually what they do is they replace the floor of the arena because the rat might have peed in a certain place, or maybe there's a little fragment of food, and it can use that olfactory cue to figure out where the location is, so you get a new floor. And then what you do is you move the cue card. So the cue card is moved to a new location, and then you just do the exact same thing again, let the animal run around. And what happens now? Well, the place field rotates. So we can be pretty sure that all we did was we just changed the location of the card, and that moved where the cell fires. So now the cell will just fire there for 20 minutes. So what is the critical second observation? So the critical second observation is that, okay, now what happens if we turn off the lights? So clearly the cue card is the main thing that is determining where this place field is. It's where, that the animal is orienting its whole sort of environment to this cue card because there's not much else in the environment. No, that's like the main visible thing in the environment. Now, what if we turn off the lights? The animal can't see the cue card in the dark. But what happens? Well, the place field's still there. We have to let the animal see where the place, where the card is. So put the animal in the enclosure so they can see where the place, where the actual cue card is. So this is, cue card. And then turn off the lights. And what happens? Well, nothing. Place field's still there. And so what does that mean? That means it's a deep observation. What it means is that the cue itself is not the thing. It's the thing that's controlling where the place field is, but there must be like a whole bunch of other stuff, in there. So obviously, the visual signal is, critical for pegging where the animal is in the environment. But if you turn the lights off, it can't see that. And so it must be using some vestibular information. And we'll see that if you look at head direction cells, which code a particular direction in the environment, the vestibular system is completely critical to their operation. But it can use auditory information too. So if you close your eyes and walk up to a wall, you can tell when you start to get close to the wall just by the different sound that, like especially if you're just making a little noise like this, and you go up to, or rub your hands together as you walk up to a wall, you'll immediately be able to tell how far you are from the wall. So you can use auditory information in the dark, but you can also use efferent's copy. So efferent's copy of locomotory signals, locomotion signals, easier. Locomotion signals. And so basically, the place cell must have information from how many steps you took. And so it has to know what direction you are, so what's your head direction in the environment, because if the place field happens to be right here, if I back into the place field, the cell will fire. If I come into the place field going forward, it will fire. If I come into the place field with my head pointing this way, it will fire. And so in order to make that happen in the dark, it's got to sort of be totaling up how many steps you took and how fast they were. So if I move more rapidly, the cell has to fire differently than if I just saunter into the place field. So all these things must be going into making the place field appear in the same place. But there's yet another thing. If you just sit in the place field, don't do anything, don't move your head, don't make any noise, keep the lights off, the cell still fires. And so there's another thing, and we'll talk. It's attractor-like firing, even when there's no cues. Even when the cues are completely off, the cell will still fire when you're in the place field. So a lot of things are going into generating this. You can see how complicated it's going to be to try to figure this out. So like the striatum gets input from the motor system, and that projects into the entorhinal cortex. That's maybe how some of the efferents copy things, get into the hippocampus. The auditory, visual, somatosensory stuff is all getting in there, through the projections from these higher level areas into entorhinal cortex. And then you've got this kind of attractor-like behavior, which allows the system to have a concept of a place, even when there's zero information currently coming in. Okay, so these are, these are place cells. And so as soon as people realized this is actually a real thing, they just went bonkers. There's, thousands of papers on this, on studying place cells. And one good one that sort of illustrates the distinction between proximal and distal cues is the use of the radial arm maze. And so basically somebody made this thing's like 6 feet, 8 feet big. So they made this big plastic maze like this, plexiglass maze. And if you look at it, like here's one of the arms of the maze. It goes back, like that. It has a little sort of like blocker on the end. And it's open at the top, and the rat is in there and just running around. So here's a little small rat. It's kind of running around like, maybe there's some food in that one or food in that one. But it's sitting in a room. So it's sitting in a room. And so if we, let me put a room behind this one. So here's the room behind this one. So here's all the other, directions of the maze. Okay, so what happens is, how does the animal know where it is? Well, it just looks out, and maybe there's a door over here, and maybe there's an oscilloscope sitting over here in the old days when there used to be oscilloscopes, and then there's maybe a little window on the room over here, something like that. So there's a bunch of distal cues, and maybe there's just like a table over here. Let's put one more distal cue in. So you got a table just sitting here, and so that's how the animal actually tells where it is in the room. It uses these distal cues, and so we call those cues that are kind of stuck down to the environment. And these cues are actually contrasted, versus, proximal cues. And what are proximal cues? They might be something like a little spot of pee in the maze. Like maybe the rat made a little pee mark here, which he can easily smell. And that's kind of a proximal cue. But it turns out if you rotate the maze, so this whole thing is kind of mounted on a little pivot point. So you can rotate the maze so that maybe this arm used to be near the table, but now you rotate the maze and this arm is now near the door. What will happen to a place field? Well, the place field will essentially be defined by what it's close to. So if this guy was, if this arm, the place field was in this arm, and then we rotated the maze, the place field will be in whatever arm is near the door. So that's the idea of a distal cue. So the animal will override the local smells inside the maze, a proximal cue, and it will use this kind of like larger scale, less likely to change environmental cues to control the firing of a place field. So this is, so the place field, it's basically near the door. It's not a particular local cue within the maze. It's just whatever arm happens to be near the door. That's where the cell will fire. So that that concept of of sort of proximal versus distal cues is going to apply when we talk about head direction cells and grid cells as well. And the number of experiments, it's hard to sort of not get carried away with all the number of different experiments, but another experiment that's been done is is called the water maze. It's not really a maze, but basically what you do is you get a little swimming pool, rat swimming pool, and fill it with water. Rats can swim. And they don't particularly like to swim. And then you put milk in the water so that they can't see where this little pedestal is in the water. So you got a little pedestal in the water, just under the water. And so what the rats will do is, if you put them in there, they'll like swim around looking for the pedestal. And so here's that little hidden pedestal somewhere in the water. When they find it, they'll hop up on it and like groom themselves and clean off and say, yeah, I found it. Yeah, I'm a happier rat. So if you allow a rat to sort of find the pedestal a couple times, and you can have a cue card. You'll have a cue card someplace. So here's a little cue card sitting in the milky water. And so that guy will tell the rat, like, where the pedestal is likely to be. Now, if you take the rat out and put him back in the water, so here we'll look at it from the top, and say the pedestal was, right here. What will happen? Well, the rat, and say the cue card is right here. What will happen is the rat will just, if you put the rat in here, it'll just swim to the pedestal because it knows where it is relative to the cue card. If you put it here, it'll just swim to the pedestal. If you put it here, it just knows where it is using the distal cues, in which case this was just like a cue card. But it's always important to keep in mind the nervous system is very resourceful and it uses multiple methods. And so when people first found these place cells, the first thing they did was, let's lesion the hippocampus and see what happens. Does the animal have trouble in a maze? Turns out, no. If the animal has learned a maze, it can still run the maze, even though you blew out its place cells. And what's going on here? Well, maybe the rat doesn't have a feeling like it knows where it is, but it still makes the right decision. And how could it do that? Well, it could use the basal ganglia or the motor cortex or the cerebellum. Maybe the cerebellum has sort of like coded some sequence of moves through the maze, and so that it knows, like, go in, make a right turn, skip the next right turn, then make a left turn. That's that kind of thing that the basal ganglia is good at, sort of sequencing things. Now, the whole time the rat might say, like, I have no idea where I am. But then they come up to a junction point, and the striatum says, I go right. And so they, what the hell, I'll just go right. It turns out their maze performance is fine. And so they can actually, so there's multiple ways of sort of doing spatial navigation. This is not the only way of doing spatial navigation. It's not even critical for spatial navigation. You can actually do it without it. But what happens if we lesion the hippocampus in this place? This is an interesting experiment. So if we lesion the hippocampus in the water maze, there's a very characteristic thing that happens. So here's the cue card. So the animal will still be able to find the pedestal, but how does it do it? It uses a different strategy. So the animal knows the pedestal is like pretty close to the edge. And so what it will do, so here's a hidden pedestal. If you stick the animal in here, what it will do is just swim in a circle until it runs into the pedestal. Because it knows, like, it's not too far from the edge of the thing, and so if I just swim around here, sooner or later I'll run into it. And so this is normal using distal cues. Use distal, but this is hippocampal lesion, hip lesion. Other strategies. So there's other things you can do. So it's important to sort of realize there's multiple ways that we can sort of solve a problem. Yeah, and like I said, when people first did these lesions, they realized, well, it was not a big deficit in spatial learning. Yeah.
You're saying that the normal or without the lesion, they'll just go straight to the little pedestal, and then they're just going to go in circles until they find it?
Yeah, this one just goes in circles until they find it. Because they can't, why do they do that? The reason is they can't relate this distal cue to where the pedestal is. The system that's sort of tied up, this is a particular place where the pedestal is, and we find that place by figuring out where we are relative to to this local cue. That system doesn't work. But they still have some information. Their motor system still knows. I know it's kind of like about this far from the. I don't know where it is. I don't know where I am in the environment, but I know it's about this far from the edge of the container. So I'm just going to swim in a circle until I run into it. So I just two last ones. So there was a great experiment where, like, what happens in 3D? Like, because when I was living in Chicago, I had like a trash apartment. There were rats on the walls, constantly climbing into the stove, and then they're climbing over by the somewhere in the bedroom. So somebody made a spiral staircase for a rat. So they made this 10-turn spiral staircase. It's like a little spiral staircase like this with made out of plexiglass. And they trained the rats to sort of like go up this, to climb all the way up the spiral staircase. And they just checked, do you have any floor-specific place cells? And the answer is yes. You'll have a place cell that fires usually on a couple floors. Say it fires on the second and the third floor, but it won't fire on the same position in the spiral. So this was the spiral maze. That was kind of an amazing one. And there's been a lot of study, if you like 3D, there's a lot of study of fruit bats, how the hippocampus works in fruit bats, because they can sort of move in 3D, flying around trees. So there's the great spiral maze. And then there's another experiment with looking at how these cues. So one of the problems is what about when the cue is actually being attached? When you haven't, like the very first time the rat comes into the maze, or into the arena, it's got a cue card. How does that happen? Obviously, it hadn't seen the cue card before, so how does it somehow attach that cue to a particular place cell? How does the place cell sort of originate? And so this was like my favorite experiment. So you got a cue card here, and the rat's in the arena, and you record the place field and say the place field is over here, like this. So there's your place field, basically. And then the rat's just in there. It's it's doing its rat thing. It's running around. So it's going all over the place. And it's not particularly paying attention to the cue card. And then you just slowly move the cue card. So let's just like slowly move this guy while the rat's kind of watching and just running around, to over here. So let's just slowly move it. Move while rat watches. Well, rat does its thing. And what happens? The place field doesn't move. The place field just actually stays in the same place. And then if you take the rat out of the arena, change the floor, put him back into the arena, move the cue card somewhere else, the place field will be in the new location relative to the cue card after it was moved. So you've actually, like in real time, you've re-specified what the relationship is between the cue card and the place field. And of course, this has to happen all the time, because you walk into the kitchen or walk into the dining room and somebody's moved the chair. I always say, a normal person would not bump into the chair if the chair was moved, and they wouldn't suddenly say, I don't know where I am in my kitchen. They would just say, okay, somebody moved the chair. So I'm just going to have to sort of update my representation of where things are in the room. So that's got to happen all the time, because there's a lot of movable objects in the world, like, for instance, other animals. And so, but here you can see it actually being re-specified. You just move it. The rat it's not particularly paying attention. It's moving all around, rearing up, sort of going like this. You just slowly, yep, I'm moving it. And then right after you finish moving it, take the rat out, stick it back in, and now you've re-specified the relationship between the cue card. So, and then last but not least, I'll talk about that one when we talk about the head direction cells. Okay, so that's place cells. But then there's other kind of cells which were discovered, and again, they could have been discovered earlier. So these are called head direction cells, and you can sort of imagine, what this is going to be. And these cells were originally found in the retrospenial cortex. So let's just describe where that is. So if you look at a rat here, there's the cerebellum, pontine nuclei, olfactory bulb. So if you look at where the hippocampus is, it's kind of sitting here like that. But if you look behind the hippocampus, in something that kind of looks like entorhinal cortex, so right behind the hippocampus, there's something, and it was confusingly originally called the post-subiculum, even because it's kind of behind the subiculum. Post-subiculum, that's where they were first found. But it's really what we would call retrosplenial cortex. That's an older word. And retrosplenial cortex means, behind the splenium, which is the end of the corpus callosum. So that's the splenium of the corpus callosum is the end of the corpus callosum. So retrosplenial cortex is where they were found. But since then, they've been found in a lot of other places. So, there's retrosplenial cortex. It turns out there's a ton of them in the mammillary bodies. So, mammillary bodies. And there's some in the dorsal thalamus also. So there's some in the lateral dorsal, LD dorsal thalamus. So that's mammillary bodies are part of the hypothalamus, but lateral dorsal is part of the dorsal thalamus, like the lateral geniculate. And there's a big projection between the lateral dorsal nucleus and the mammillary bodies. That was the mammillothalamic tract that I mentioned last time, lateral dorsal thalamic nucleus. So there's a lot of them. There's a lot of places that have these head direction cells. And what do these look like? They look like compass cells. They just look like an internal compass. And so if you actually look at, if we make our arena here, we have some cue card someplace in the arena. And what do the cells respond? Well, every year I draw this, but I'll try to explain it a little bit better. So we've got a rat in there, and this rat is an improved rat. It has like two headlights on it. It's got like a red headlight and a green headlight, so you can tell what direction with the camera the head is pointing in. So there's the red and the green headlight, and the rat's just running around like it normally does. And now what you do, it's a little more complicated. We have more variables. We've got X and Y, because the rat can be in different positions, and then we have the head direction, what angle the rat's head is at. So there's three variables to consider. And so if we just record for a half an hour, we'll find that the cell basically will fire when the rat's head, no matter where the rat is in the environment, it will fire when the rat's head is pointing northwest, like that. So that's what those arrows mean. So those arrows mean we bend things by where they were in X and Y, like we did back over here. Spike is a function of position, X, Y. So we bend them that way, but then at each XY position, we also bend them by what direction. And then so like at this XY position, we look at all the rat's head direction and just make an average arrow that indicates what is the head direction that caused most firing. And so if we just plot that, again, like when you first see these cells, it's just like, could this actually be true? So here's what a graph looks like. So here's the response. This might be 50 spikes a second, spikes per second, and you get a response that looks something like this. I mean, it's just unearthly. I'll make it sort of like, so say at 45 degrees. So basically it'll just come up like this and then go down like that in completely free movement for a half an hour. So that's 0 and 360 head direction. And so how did people find this? They actually found it. They were the people who found it, Jim Rank, Jim Cuby, one of them is John Cuby and Jim Rank. They were trying to record from the hippocampus. So the hippocampus is kind of sitting right in front of the postsubiculum or retrospenial cortex, and they were just in the wrong place. And they often have the neuron hooked up to a speaker so you can hear the response of the neuron. And they just picked up the rat, and these rats are very used to being handled. And so they picked up the rat and just like said, like, look at this, John, it looks like a compass cell. And so they're just holding the rat like this, and they just go like, turned it around like this, nothing, just an actual compass. And so they said, well, I don't know, it just seems to be responding. So they took the rat into another room, and sure enough, in the other room, still had the same compass direction. Now, it's not using actual magnetic fields to do this. It's just using the cue cards, just like before. But it's just incredible that this could be the actual neural response of recording from a cell for a half an hour while a rat is running around an arena and sort of lifting you're rearing up and chasing down chow and running and backing up, and you get something that looks like this. And then if you record another head direction cell, you'll see, another curve, somewhere else like this that responds to a different head direction, about that sharp. So it's unbelievable that this could actually be true, but it is true. And what happens if we do the standard experiments? Exactly the same thing that happens with place cells. So we can do this kind of experiment, just bin spikes by where the location is, and what happens in that case? Well, the spikes are all over the place. There's no location sensitivity. The cell, it's not like a place cell, it just responds wherever. Similarly, if we take a place cell and do this kind of experiment and just record their head direction, there's very little effective head direction on the place cell. So like the place cell will fire if the rat is aimed at this direction, or its head is aimed in this direction, and head is aimed in this direction. It doesn't matter. The place cell fires. Whereas these guys, they require that the rat's head direction is in a particular direction. But if you do that same kind of experiment where you just move the cue card, what happens? Well, the head direction just changes. And so if you start off, with your head, if you put the rat in here, record for a half an hour with the cue card there, pull the rat out, confuse the rat, stick it back in with the cue card over here, what happens? Well, all these head direction cells, will now rotate. So it's important to remember they're not pointing at the cue card. They're pointing in a compass direction. So a lot of times the students get mixed up that they're not actually pointing at the cue card. They're just pointing at a particular compass direction as specified by this cue card. And so it works the same way. If you turn off the lights, still works. If you turn off the lights and the animal's just staying completely still and happens to be in the head direction, the cell fires. So, like the attractor type firing is just causing it to fire even though there's no cues coming in. Then there was the earthquake experiment. So this one, so the rat is just like, so how are these things coordinated? So these things are all sort of coordinated with each other. So they're, we have, coordinated, inertial guidance system. you'd guess they were coordinated because if you rotate the cue card, the head direction cells rotate. You rotate the cue card, the place cells rotate to the new place. But this can happen in a very short period of time. And so they did this experiment where the rat was in the maze like this, and it had the cue card someplace. So the cue card was someplace, and you recorded some place cells, say here's a place cell, and you recorded some head direction, like recorded a head direction cell, and the head direction cell, was pointed sort of like northwest there. And then what you do is earthquake. And so like you just come in and like the rat's just minding its own business, and you just like jerk the whole arena around. And what will happen then? So then what will happen is the rat, not all the time, but if the rat is maybe disturbed enough by this, it resets its. So you just do an earthquake. What happens is the cue card hasn't moved, the arena hasn't moved, just got jerked around a little bit. But what happens is it will update all the place cells and the head direction cells in concert with each other to some other random location. So what will happen is now this place field will rotate around to over here, and then these head direction cells will all rotate with it. So the whole thing, this whole system is like it's one big coordinated system that's representing where you are in the environment. And the important part about this is that it essentially has a concept of being somewhere that doesn't require any data. It's like you're always somewhere. I'm somewhere. I don't know exactly where I am, but I've lost all the cues that tell me where I am, but I'm still somewhere. So the system has an idea of there is one head direction that your head is in, and there's one place that you're in. And all the place cells and all the head direction cells will respect that and sort of operate as a coordinated system. So the place cell system and the head direction cell system have to be closely tied to each other, even though they complete a different response. The head direction cells could care less about where the place is, and the place cells could care less about where the head direction is. So that's the and then one last favorite experiment. So there's another experiment where they got a rat, and they made one large arena like this, and say you had a couple cue cards in it, and you put and there's a little door, secret door here, and then there's another arena that's attached to it. And say this one has some cue card here. And so you separately put the rat into the first arena, and the head directions might be, that direction, like that. And then you don't allow them to go through the door. And then on a different day, you put them into this arena, and the head direction is not controlled by magnetic fields, and so the head direction is some other direction like that. And then one day, when they're in the first arena, you open the door and keep recording the head direction cells. So here's the first arena, second arena, and so the rat goes through the door, and then when it sees this other arena, it says, "Oh my, I'm not in the right place." And what happens? The head direction cells immediately rotate to the new orientation, compass direction, that's appropriate for the one they learned in this room. And what is that like? Well, if you're not very good at navigation like me, or especially if you're in the car and you're driving around it's a very slow turn, you don't realize your vestibular system has trouble sort of integrating a very slow turn. If the road sort of very gently curves for a long time, it's hard for your vestibular system to pick that up. And then you come up to an intersection, and then suddenly you recognize that intersection. You go like, "Whoa, man! I thought that was north, but this is north." And all your head direction cells are going sort of rotating around to the new cue. So once again, you have all these different sub-environments, and they have to be sort of knitted together into some kind of coherent picture. And so you can see, you can see the rat's head direction cells just rotating in real time as they enter into the new container, and they realize, oh, that's the cue, so that's actually, the real position of north. So these are kind of almost more amazing than the original ones, like, how could, does the brain actually work that way? Yeah, there's tons of them. They're all over the place, head direction cells. Okay, and so last but not least, the grid cells. And so, again, these things could have been discovered right at the very beginning. They could have been discovered in 1980. They had all the technology to discover these in 1980. And really, what it was just a failure of theory. People couldn't believe that the brain actually works this way, even though it does. So what are grid cells? So grid cells basically have they fire on a hexagonal grid. And, again, it's like, could that actually be true? So here's an arena. Again, it's such a strange thing that nobody ever looked for it. And the people who did look for it or just sort of recognized it when they were looking at their data, they got the Nobel Prize for this. And so here's a hexagonal grid. So we just make a hexagonal grid here, like that. So there we go, good hexagonal grid. And so what does this cell look like? Well, you just put the rat in there, you got a cue card someplace, and the cell just fires at these grid points. Can you actually believe that's actually the way the brain works? Again, this is like the brain really doesn't work this way. But if you do a half an hour of recording, you'll see something like this. It's unbelievable, but it's true. And where are these guys located? They're located in entorhinal cortex. And so where is, entorhinal cortex? So let's draw a slightly bigger rat. So here's a bigger rat. Olfactory bulb, cerebellum, pontine nuclei. And so where is this? So if you look at the hippocampus is kind of sitting something like that. And then we said the entorhinal cortex, it's kind of right next to the hippocampus. And from a side view, it's slightly behind, like that. But it's parallel to it. It's about as long as the hippocampus. And so this is entorhinal cortex, and this guy is hip. And so that's one of the things that provides input to the hippocampus. But remember, it also gets output. The entorhinal cortex also gets output from the hippocampus, so it's a little hard to figure out like what the order is. But these grid cells, were, in entorhinal cortex. That's where they were found. And one interesting thing that was found about this is that there's a correlation between the size of the grids in different parts of the entorhinal cortex. And so if you go to the, I think I actually got it wrong. I put it in the wrong order last time. So if we, is it large, small? Where is it? I know small is at the top, but yeah, okay. So if you look at the grids, so we get small grids at the top in entorhinal cortex, and we get small place cells at the top. No, I did it right last year. Small place cells at the top in the hippocampus. And if you go deeper into the entorhinal cortex, you get large grids. So you get large grids here, and you get large place cells. And one of the reasons, large place cells, one of the reasons they didn't notice this at the beginning is that some of the place cells are really big. And I always tell this as an example of why I need more lab space. The best argument for more lab space, because I've got to find these grids that are large grids. And so the Norwegian guys who figured some of this out got this like giant 40-foot lab space, and they made a huge arena for these rats. And you can see these rats galloping across the arena. And they found these big place fields. They're like 5 feet across. So they're really large sort of large place cells, but also larger grids. So the spacing between the grids gets larger. So it's just like, could this be possible? And so people have suggested, well, maybe you can make place cells by overlapping a bunch of grid cells. That's one idea. Some of the place cells develop before grid cells, so that's a little hard to reconcile with that. But there are definitely tons of these, and this has been now repeatedly shown. There's tons of work on grid cells. So it tiles the environment with these firing rates that are just sort of, it's kind of hard to imagine, but as you walk by, all your different grid cells are firing at different times as you hit these little grid points in the environment, Just incredible. So how could this possibly work? How could something like this possibly work? So there are several models for this. So one model has to do with the theta rhythm. And so the theta rhythm it's like say 4 to 7 hertz cycles per second, rhythm that happens. In rats, it happens on every breath. So when a rat is sitting there and sweeping its vibrissae, it's going, it's making these constant sniffs. And if you look in the hippocampus, you can see this rhythm that's in sync with the sniffs. Now, other animals don't sniff as rapidly, but they still have a theta rhythm. So it's not just due to the sniffing, but it seems to be coming from the septum. So the septum has these driver cells in it that sort of have the theta rhythm. And the theta rhythm isn't there all the time. Sometimes, especially if the animal's not paying attention, it goes away. But when the animal's kind of doing some active exploration, you'll see the theta rhythm. And so then people looked at does the cell spike during the theta rhythm? Yeah, it tends to spike during the theta rhythm. And one thing they found was that there's a change in the phase of the spiking as the animal goes through the place field. So you can see that the theta rhythm, the spikes will sort of occur in a different place in the theta rhythm cycle as the animal goes through the place field. And that suggests that maybe there's some kind of interference mechanism with multiple slightly different theta rhythms that are involved in making the place cell. So that's one idea. That may be partly true. But another idea that we're going to briefly talk about is an attractor network. And it turns out if you let's just start with head direction cells. So let's make a circle of cells. So here's a circle. Sometimes we call this a line attractor network. And you'll see why we call it a line. So here's a circle of cells. It doesn't have to be an actual circle in the brain. It could be like, they could be all jumbled up as long as the connections respected a circle. And if we put, positive connections to nearby cells, so that's plus, and then make negative connections to farther away cells. So, and for the start, let's just make negative connections to all further away cells, okay? So these are all negative. So inhibit everybody else, and just excite your neighbor. So if all the cells are like that, so if you make all the cells like this so that everybody excites their neighbors and everybody inhibits the people that are further away, what happens? Well, that's going to be an attractor network. It's basically going to be a winner-take-all attractor network. And so what will happen if you look at, so let's just draw the ring of cells like a line. So what will happen, say you start the state, remember we were talking about different states, which means different activations for each of the units in the network. Say we start off the state of the network in some, the height of this little mountains are like, how active the cells are. So say we start off the state in some random looking state like that. So this is our random starting point. So we just make a network, hook up to your neighbor's excitatory, hook up to everybody else inhibitory, do the same thing for all the cells in the network. So this is our starting state. What will happen? Well, what will happen is wherever the randomly slightly highest activation in the ring is, you're going to get a winner-take-all because these guys will slightly inhibit everybody else a little bit more. And so what will happen is you'll end up with basically, zero activation and then a little bump of activation over here like that. Then everybody else is zero all the way around the ring. And so that's a stable state. But it turns out you can make a stable state anywhere. And how could you do that? You could do that by, say, what if we just stimulate these cells here a little bit? So just some outside stimulation comes in and just stimulates these a little bit. What will happen? Well, you'll move the bump over there. Like that. You just actually move it. So there's always one bump. There's always one bump, but it's always someplace. And that's just a consequence of this simple attractor network dynamics, which is that if you've got an attractor network like this, you have all these stable states. Now, in the attractor networks we talked about, a lot of times the attractors were, like we imagined, here's our energy landscape, and we've got something like this. And if I start off here, I'll go down to this attractor, but if I start off here, I'll go down to this attractor instead. So, here's attractor 1, here's attractor 2. This one's a little different in that essentially the attractors are kind of infinitely close to each other. So we could have, if we just slightly tweak this guy with a little more stimulation on this part of the ring, we can just move this bump just over, So basically there's like a million, infinite number of attractors all the way around the ring. But the important thing is they don't need any input. They just do their thing. And so as soon as you get the state of this ring of cells into an attractor, it doesn't need any information from the vestibular system or the visual system or whatever. It's just I'm pointing this, my head is north. And I lost all the information from my vestibular system because I'm not moving my head. If somebody turned off the lights, I'm not moving my ears, and so I'm not getting any information about how far I am from the wall, but I know I'm here. And how could you sort of integrate all the cues into it? Well, there's like a zillion things coming in here. So, here's a cue card, and then here's what the wall looks like when I'm looking the opposite way from the cue card, and here's some vestibular input that indicates I should rotate these things. So there's a ton of inputs coming in here, but it's got this internal structure in its firing pattern, which is to always make a bump in a particular direction and always have some concept of what direction am I pointing, and then also talk to all the other place cells and head direction cells and cause them to all rotate in concert with each other so that you've got this coherent representation of the environment that is so strong that even when you cut off all the information, it's still there. The whole thing is still there and still organized. So that's one key way in which these attractor networks might help us sort of conceptualize what's going on here. So you've got a system that makes a bump, like for the head direction cells. It makes a bump somewhere around this circle of cells, and then all different kinds of information can come and push the bump around: vestibular information, navigation information, muscle information, visual information. But it's not required for the cell to keep firing. It'll just fire, once you stop, it's still firing. It's still telling you this is the direction your head is in. This is where you are in the environment. So these kinds of models, perhaps put together with some of these theta interference models, might be just the beginnings of how this can work. How the whole thing is coordinated is nobody's come close to actually sort of trying to figure out how does the vestibular input come in and push the bumps around. How does the visual information come in? Where are the synapses that change when we add a cue card or remove the chair in the dining room? Where are those things actually? Are they in this piriform cortex? Are they somewhere in the striatum? Who knows? We don't really know. But this is just the beginnings of an idea, of how this thing might work. And in the last 1 minute, there's still 1 minute before the hour is up, we could talk about what if we did something where instead of synapsing, like we could do the same model for a place cell. So a place cell, so here's a place cell, it might have excitatory projections to its neighbors, and then inhibitory projections to ones that are further away. Now, if we inhibit everybody else, then you'll get the same winner take all, so that the stable state will be a little bump somewhere in this network, that this little bump, that sort of bump of activity in this sort of two-dimensional map of cells that moves around. And what moves it around? Well, the vestibular information came in, efferent's copy came in, information about where the visual cue is came in, and it could maybe move this bump around. But if the inhibition doesn't go all the way out to the whole rest of the network, you could get multiple bumps. So, there could be another bump over here. So you could have sort of multiple bumps moving around in the network. And that might be possibly a model of how the grid cells work. You're moving multiple bumps around. It's just an idea. It's just an idea for how it might work. But there's no question that this phenomena exists. The place cells, the head direction cells, the grid cells, and there's other ones I haven't told you about. So it has to be eventually explained. So I think there's another 10 experiments we could talk about, but I think I'll just end it there and see if there's any questions. So any questions about this? I should label this. This is 2D version of ring. So this is 2D version of ring, line attractor network. This is a 1D ring. Yeah.
The attractor network only applies to the head direction cells, or what mammillary cells as well?
No, this could be an attractor. Yeah, so we can label this. This is like attractor model of place cells. So if you make an attractor network where each unit in the network excites its neighbors and inhibits everybody else, and then make all the other ones the same, the stable state, instead of just like a bump on a ring, it will be like a bump on a two-dimensional field. And so if some other information comes in, it can sort of move the bump or cause a bump to appear in a different place. So if you take that network, you start it off in a random state, wherever the highest random collection happens to be, that thing will sort of like grow up into a bump and everybody else will go down. But then if I just stimulate some other part of the sheet with enough input, maybe slightly more than that other bump was getting, I'll force that bump down and make this bump come up. And then if I stop stimulating, bump stays there. So it doesn't require the stimulation, but the stimulation can move it around. And so it's the same kind of thing except in 2D, where when we were talking about the head direction cells. So this is, this guy is, for place, and this guy is, for head direction. So head direction is only 1D. But whatever system moves stuff around must somehow work in both of them. It has to work in both of them to be able to cause both of them to change around. So isn't this amazing? Can you believe the brain actually works this way? But
What if you have like two signals or stimulus that's like of the same strength, and you kind of stimulate at the same time? Would it also like still have that bond?
no, that's a good question. So if one cue says like it's definitely, this is north, and the other one says no, this is north, if they're exactly balanced, you can temporarily maintain two bumps. But as soon as one gets a little stronger or slightly less stronger, it's winner take all, and then one will go down and the other one will pop up. Yeah, and so it would have to be some kind of cue that was exactly balanced. But basically what happens is if you just randomly stimulate the thing, you'll always have one bump. You'll just shove one bump around. If you're inhibiting everybody else. If you're just inhibiting neighbors, then you can have this multiple field of bumps. Some similar things could be going on in the cortex. You could easily imagine multiple bumps kind of propagating around the cortex, because the cortex tends to be more sort of excite neighbors, inhibit slightly further away neighbors. Guys that are really far away don't connect to them. So, so you could easily have multiple bumps going on in the cortex. Okay, so these are these amazing cells. It's very good, it's a good thing to look at, because people did not think the brain works this way. I did not think the brain worked this way. And this is something that the brain has told to us. We actually did some experiments and we realized, wow, it doesn't work the way I thought it did. That's really valuable data. But it also shows why it took so long to find things like the grid cells. They could have found them in 1980. Could have gotten a Nobel Prize in 1980. Okay, any final questions about the amazing place cells, head direction cells, and grid cells? All good? That's a good question. So how do they stay active? They basically just stay active by these connections. So imagine you're in the state of the bump being here. Then what's going to be happening? These guys are going to be exciting their neighbors. That keeps this side of the bump up. And then they're going to be inhibiting everybody else. And so it keeps all those guys' bumps down. And if they're down, they can't inhibit you. And so you just have like all the friendly excitation from your neighbors. But as you go further away, some inhibition starts to kick in from this central guy, because the central guy will be inhibiting these guys. Now these guys will be being excited by their neighbors, but they're going to get a little more inhibition than the central guy will. So that's why the central guy will be the highest, and the ones next off to the side will be slightly lower. And so all it needs doesn't need any input at all. It just needs that this guy is being excited, and because this guy's being excited, it's exciting its neighbors. They're in turn exciting it back, and then they're inhibiting everybody else. And because they're inhibiting everybody else, those guys can't come back and inhibit you because you turned them off. So it's basically a winner-take-all type thing, where you start off with who's ever the biggest gets goes up to maximum and everybody else gets turned down. Yeah, so to keep it going, you need like, this guy has to be continuously firing. But we know that that's actually the case. If you just hold the animal, if the head direction cell is straight ahead like this, if you just go, if you just hold it, it'll just sit there and just fire continuously. So I mean, what's happening actually in this ring, as you move your head around, the little bump is just going, wah, 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 wah. You can stay in one place for as long as it needs to, but as soon as you rotate your head, somehow the vestibular information comes in and pushes the bump around. What's that? Yeah, well, it's kind of like it's got a sense of direction, even when there's no data. I think that's the important point. It's kind of like the way that you have a sense of a category. You have some category representation of a dog. You're not seeing a dog, but you can sort of like think in your head, oh, I'm just thinking of a sort of a generic dog. I can think of a generic dog, and it doesn't require an actual dog to be there. And that's kind of the same thing here. It doesn't require an actual head, any input from the vestibular system. It says like north. It's just north. Yeah, I mean, how can they get turned up and upside down? Yeah, so if you're underwater, you're almost in this Ganzfeld sort of like uniform field. It's very hard to see which is up and down, and you lose sense of gravity because you don't feel you're more or less floating, and so you don't feel like which end is down, and it's quite easy to get disoriented. And you could imagine that's kind of like the earthquake situation where you go like, I don't have any idea where I am. Like, let's just randomly rotate. It's kind of like when I walk out of a store. I walk into the store, I come into the store, turn left, go into the store. And then I walk out of the store, and half the time, instead of turning right and going back to where I came from, I turn left. And I figure it's like the earthquake situation. Like, I have no idea where I am. So let's just go somewhere else. And so instead of like turning right, I turn left because it's not right. So I think this system it's like a coherent system, and it can, under certain circumstances, it can be destabilized. But how does it destabilize? It doesn't just become random. It just like rotates to a coherent new position. And this can happen, it can happen when somebody's flying. Like if you fly into clouds, what can happen is you lose all kind of like visual orientation, and vestibular information detects changes, but it doesn't detect static position that well. And so what can happen is you can slowly tilt your plane, and you're just flying. You feel like you're going straight, and then you come out of the clouds and you're completely upside down. And you go, whoops, that's not right. Let's turn it back over. So the bottom line is that this whole thing is a giant coherent set of attractor networks that are all coupled to each other, so that there's always like a coherent representation of the whole environment. It's just like it's maybe not stuck properly to the environment anymore, like if you go underwater and you lose a lot of the cues. So you can get off if you take away all the cues. Well, so in the case of rats, if you just lesion the vestibular system, the head direction cells don't work. So that's the most direct way of doing it. But you could easily imagine the same thing in a human. If you have some vestibular damage, then you can have much greater problems sort of maintaining, keeping this system running. But there's a lot of other overlapping cues that can sort of help. I mean, everything goes in there pretty much. But yes, definitely in the rats, if you lesion the vestibular nucleus, the head direction cells get completely messed up. So we know they're relying on every time you rotate your head to rotate the ring around. Okay, I think we'll end the recording there if it's not already turned off, Stan.